You push that to start.
Okay, okay.
See it's red, that means it's filming. And then that zooms out and that zooms in.
Okay.
So
if I want to point it up at the board, do
here we
I
go.
loosen this?
Yeah,
and then...
you can loosen that and move that around and tighten it.
Okay.
Whatever you want to do, or you can just hold it up.
Okay, hold
make
on.
sense?
Yes.
She's probably not the best one.
Dude, are you kidding me?
Okay, so we're talking about proteins now. You know what my nickname was when I was a teenager?
Protein? What?
Protein.
Why?
So I'm protein.
Oh,
Like
I
professional.
get it. I get it.
Oh, that's a good spot.
That's a joke.
Oh crap.
You got it?
There. Right there.
Please watch your language.
Oh.
Okay, what's a protein? A protein is made of individual units called amino acids. Can you say amino acid?
Amino acid.
Very nice. Amino acids are joined together in a long chain to make a protein. So
That's easy.
this string of beads is like what a protein structure is. But each little bead is an amino acid, and they're all joined together in a long chain. Now you need to know the structure of an amino acid. You have to know that. So I'm going to write it down on the board here, and you try and learn it right now, okay? There's a carbon in the middle. That's easy enough. And how many bonds does a carbon have?
4.
4 bonds. So it's going to bond to 4 things. A hydrogen, pretty simple. On one side is an NH2 group. You know what we call that NH2 group? Anyone know? It was on the last test. It's a functional group. No one?
Wait. Hydrogen? No.
What'd you say? No one? It's called an amino group. Is that what you said? No. It's called an amino group. NH2 is an amino group. So that's sticking off one side of the carbon. Off the other side of the carbon is a COOH group. That's also called a carboxyl group. It's also called an acid group. So amino group on one side of the carbon, acid group on the other side of the carbon. Can you see why they call it an amino acid?
Yep.
And hanging down from the carbon is something we call an R group. Now the R group is variable. Do y'all know what a variable is?
We should know this.
Y'all don't know what a variable is?
Isn't it something that may or may not change?
Yes, it can change. Very good.
I'm smart.
It can change. The R group can change. The R group can be one of 20 different things. So the R group could just be a hydrogen. Maybe the R group's a hydrogen. Or maybe it's a carbon connected to 3 other hydrogens. Or maybe it's a carbon connected to a sulfur atom connected to a hydrogen. It could be all sorts of things. It could be a carbon group connected to a ring structure. There's 20 different things the R group can be. 20 different kinds of amino acids. Yes.
Are we going to need to know some of the different...
You are not, but there is a picture in your book that shows them all. You want to see it?
Yes.
This shows all the different types of amino acids. So, here's the amino group. Here's the acid group. There's the center carbon. And this long thing is the R group. So that's an R group, that's an R group, that's an R group, that's an R group. And you got to understand how scientists write write organic molecules, instead of putting the carbons, instead of spending a lot of time writing the letter C, they'll just do a zigzag. That's short for a string of carbons attached to hydrogens. So y'all remember like this fatty acid tail, right?
Yes.
If a scientist wanted to draw that really quickly, they would just do a zigzag line like that. And that would stand for that. And you would have to assume that at every point of the structure is a carbon. That's how scientists, that's kind of a shorthand for scientists for drawing organic molecules. So can you see now, if you look at this chain here, that's a carbon chain with a nitrogen on the end? Here's a carbon with a double ring structure on the end. That's called a benzene ring, that thing right there. If you take organic chemistry, you learn all these structures and such. I'm not going to test you on it. You don't have to know all the different types of amino acids. You just have to understand that there's 20 of them. And they all have different what? 10 points if you and your partners can tell me the name. There are 20 different amino acids, and each amino acid, each of the 20 kinds has a different blank. Fill in the blank. I'll give you a hint. I just said it a little bit ago. I don't know if that's much of a hint, but there are 20 different amino acids. The amino acids differ from one another in their blank. White, I'll give it to you.
Woohoo!
Orange, I'll give it to you. Nobody has yet got the word that I'm looking for. Yes, pink got it. Yes, blue got it. Anyone else? Green? Green is incorrect. Anyone else? They differ in the... I almost said it. So close. Purple got it. They differ in their R group. Their R group. Let's go back and look at it. See that huge R? What's the deal with that R?
It can change.
It's variable. It could be one of 20 things. So when we do our model, do you see all these little circles here? Each circle is an amino acid. And they can bond together to form a long string. And there's a special name for a bond between two amino acids. It's called a peptide bond.
400?
Let's take a look at how a peptide bond works. Here's an amino acid here. Can you now tell that's an amino acid?
Yep.
How do you know it's an amino acid? If you're looking at that, how would you know that's an amino acid?
It has amino on one side and acid on the other.
How good an answer was that? Who said that?
Anna Grace.
Anna Grace. I'm impressed.
Good job.
She knows that this is an amino acid because it's got amino group on one side and acid group on the other.
What is it called when there's a bond between two different?
That's what we're going to get to, a peptide bond. So let me show you. There's an amino acid there. Let me make another amino acid over here. I'm drawing another amino acid. Look how fast I drew that amino acid. I'm so good at drawing amino acids. Now, if we bond these two amino acids together, we can form a special bond. We can remove this OH group here, take that off. Remove this hydrogen here, take that off. You know what we get when we combine H and OH? H2O, water. Very nice. So we remove that OH and remove that H, and we'll form a special bond between the nitrogen and the carbon. Boop. We have a name for that bond. It's called a peptide bond. Can you say peptide bond?
Peptide bond.
All at the same time? All at the same time, louder?
Peptide bond.
Much better. So this thing right here, this is an amino acid. And here's another amino acid. And they're held together by a peptide bond. That's what these little circles stand for. So that circle there is like this. And that circle there is like that one. And they all bond together, you see, and form a string. Isn't that great? And I want to represent the string with a string of beads that I made a student who had detention paint. Why did that student who had detention paint them all different colors?
What, they painted it?
Yeah. Paint.
Oh gosh.
Sounds kind of for fun. For detention.
For detention.
Why did we do a different color?
Because I told them to. But why would I tell them to do such a thing?
Because
What do the different colors represent?
different
Different.
Different amino acids.
That's
Do it, Thomas. Different amino acids. And each amino acid has a different R group. Okay? So you have this long chain. So here's what I want you to do right now. I want you to try something. I'm going to give you a little quiz. But it's not going to be graded. You can grade it yourself. Here's what I want you to do. Without cheating, without looking at the answer, take a little sheet of paper, or you can do it in your notebook, just somewhere. Without looking, though, take a fresh sheet and see if you can draw the structure of an amino acid.
Oh dear.
See if you can do that right now. I'll give you a start. I'll give you a head start. It's got a carbon in the middle. And remember that carbon has 4 bonds. See if you can draw the structure of an amino acid. If you've been listening well, you might be able to.
Wait, we can do it in our logbook?
Yeah, just do it somewhere. I'm not going to grade it. I just want you to see if you can do it. Yeah, just draw it.
I forget what's over
Now
here.
get ready to smile when I show you the answer.
Okay.
Hold on. Okay.
You're smiling already.
Yeah.
I like that. Can you draw an amino acid? Carbon in the middle. Did you start with that?
Yes.
There's a hydrogen attached to that carbon. Did you get that far?
Yes.
On one side of the carbon, there's an amino group. What's an amino group?
And nitrogen.
N with 2 H's. How many got this far?
I did.
Crap.
E.
What's on the other end?
Carboxyl.
A carboxyl group. C double bond O, OH. That's a carboxyl group. And what hangs down? An R group. What's the deal with the R group? If you drew that or something like that, give yourself a pat on the back. Good job. If you didn't, memorize it now so you can get it. If I showed you a picture of that on a quiz, you could say, hey, I could say, hey, is that a monosaccharide? Is that a lipid? Is that an amino acid? Or is it a nucleotide?
An amino acid.
There you go. You see, you would have
Have
got
you
it
learned
right. Now, let's see, because we're not done. This chain of amino acids doesn't just sit there like this. It doesn't just float around inside your cells like that. It coils up into a final shape. It coils up into a final shape. And how does it do this? We call this the primary structure of a protein. This chain of amino acids, that's your primary structure. But what it's going to do, it's going to coil up into what we call secondary structures. There's a little secondary structure right there. You know what we call that? That's an alpha helix. How about that alpha helix? See how it's kind of coiled around?
Is that the final
Uncoiled it,
shape?
y'all. I'm going to coil it back. It's fun to do. That's an alpha helix. Doesn't it look kind of like that picture of the alpha helix? This thing right here, alpha helix. It can also form what's called a pleated sheet. What's a pleated sheet look like? Let me show you. I'm going to make it form a pleated sheet. Are y'all watching? Pleated sheet. Here we go. It goes back and forward. There we go. That's a pleated sheet. Can y'all see it? Check it out. I got a little alpha helix and a little pleated sheet there. Same string. We call these secondary structures. They stick together. The pleated sheet is held together and the alpha helix is held together by magnetic attractions between the amino acids. The amino acids can form little what we call hydrogen bonds. Little bonds between the amino acids, between a positively charged hydrogen and a negatively charged oxygen on a different amino acid, and they can form a little weak bond. And that holds them together in this little coil. So this protein now has, it's not just a long chain anymore. It's got a little alpha helix in there, and it's got a little pleated sheet in there. It's taking on further structure. It's becoming more complex. Isn't this what I have here a little more complex than just a long chain? Isn't this right here more complex than just this? Because it's folded up a certain way. But it can fold even more. It can fold where the alpha helix sticks to the beta sheet and forms a strong bond between the two called a disulfide bond. You want to see how a disulfide bond works? Disulfide bonds are some of the strongest bonds. So check this out. Do you see, where's the sulfur atom? Where's my sulfur? See that S right there? That S is sulfur. And sulfur, if you get one sulfur near another sulfur in your chain, so let me go again. Here's my alpha helix. And here's my pleated sheet. And let's say the blue one has a sulfur on the end. And if that thing gets near another blue one, the two sulfurs will stick together and form what's called a disulfide bond. And man, that bond's strong. And it makes it so this structure stays all folded up. And now we have a blob that we call not primary structure, not secondary structure. You know what it's called? Tertiary structure. That means a third structure. Primary structure is a single chain. Secondary structure are helixes and sheets. And tertiary structure is when the two of them stick together to form a nice blob.
Oh, what's the bond called?
Disulfide bond.
Can you spell that on the board, please?
Yeah, heck yeah. Disulfide. Disulfide bond. That means two sulfurs connected together. It's a chemistry term. And so now we have this tertiary structure, which is a protein that's formed kind of a blob shape. And guess what? It can bind to another tertiary structure from somewhere else. Watch this. If I have a tertiary structure over here and a tertiary structure over here, watch, they come together, boop, and they combine together and form strong linkages between the two. And man, that's a protein. That's what a protein is. Yes?
Is the primary structure just the line?
The primary structure is a long chain of amino acids, yeah. The secondary structure is alpha helixes and pleated sheets. And the tertiary structure is when that kind of bends together. And the quaternary structure is when two tertiary structures come together. And that can form a protein. And proteins help make up cells. They help build cells. We're going to talk about proteins all year long. We'll say this is a protein, that's a protein. I want you to know the structure. Yes?
So can proteins only be formed by quaternary structures?
Tertiary, what we say it's a protein. When we talk about a protein in your body, it's either tertiary or quaternary.
Okay.
Sometimes it's only one chain, but sometimes it's a couple chains together or more. Have you ever heard of a protein called hemoglobin? Carries oxygen inside your blood cells. Each blood cell has 1 billion hemoglobin molecules in it. And hemoglobin is a protein. Yes, Anna Grace.
What's the squiggly one called again?
This one? That one? I'm not sure. This one?
No,
That one?
I don't know.
I don't know either. I'm not sure what you're talking about, squiggly.
It's like,
The one that goes like this?
yeah.
That's a pleated sheet.
Okay.
This one like this? Like this when I go there?
Yes.
Yeah, that's a pleated sheet. That's a secondary structure. Are y'all getting this? All the structures of a protein?
Yes.
And this is cool. Did you know a protein can become unwound if it gets hot enough? Do y'all remember I told you that heat, the hotter a molecule is, the faster it vibrates?
Yes.
Well, imagine we have this protein and then you start heating it up. Let me show you what happens when the protein starts heating it up. You getting this on camera? This is great. Protein starts heating up. Protein starts heating up. And it comes apart. Let me show you again. Protein all coiled up, just sitting there nice. If you start heating it up, it'll denature. That's called denature. It'll come all the way apart and go back to its primary structure. And it no longer has the same look as it did before. This is why eggs change color when you cook them. They go from clear and kind of snotty looking to white, don't they? You denatured all the proteins. You straightened them all out and it looks different. When you cook meat, it goes from pink to brown. That's because the proteins are denatured. All the proteins when you're cooking come apart. Isn't that cool?
Yeah.
Curling iron, your hair is mostly protein. If you take a curling iron and make it hot, the hair's proteins will come apart and will change shape to the shape of the curling iron. See, your life starts to make more sense when you learn biology. Because it's the study of life. There's a cool looking protein. Look at that thing.
Looks like a birthday.
Can you tell that's an alpha helix there?
Happy birthday.
Oh yeah.
Here's another one. There's this primary structure, and there it is folded into its tertiary structure. That's an alpha helix. That's a pleated sheet right there, those two lines. If you ever see something all curvy like that, it's probably a protein. Is it a 225 bell or a
225.
225? Sweet. Let's see this video on proteins. You please.
Hi, it's Mr. Anderson, and in this video I'm going to talk about proteins. Proteins are incredibly important because that's what we're made up of. When you look at me, you're looking at my proteins. We just completed the Human Genome Project and we figured out the DNA in a typical human, but now we're headed into what's called the Proteome Project, where we try to figure out what are these proteins made up of and what do they look three-dimensionally. This used to be an incredibly hard process. This is John Kendrick here putting together a model for myoglobin, and you had to do it by hand. We now do a lot of this with computers. But you can help.
Watch the
At the end
video.
of this video, I'm going to show you a program called Fold It, and you can actually build and fold proteins that are going to be used in scientific research. And so it's a really cool thing. And it's a really cool time in reference to proteins. But let's start by building a little bit of knowledge. Proteins are made of amino acids, and most 7th graders understand this. They're the building blocks of proteins. But where do we get those amino acids? We get them in our diet. And so basically we eat proteins, we break them down into amino acids, and then we can weave those back together again into the proteins that make us. And so when you're looking at me, the amino acids in my skin used to be part of my food. And so I literally am what I eat. Now here's 5 different amino acids. There are a total of 20 that we use in life, but here's 5 basic amino acids. When I show this to my students, they tend to get a little bit overwhelmed because there's too much chemistry here on one page. But let's try to figure out the things that are the same on this page. And so basically, if we were to look at the middle of each of these, we find that there's a carbon with a hydrogen attached to it. We call this carbon the alpha carbon. It's going to sit right in the middle. What else is the same in every amino acid? Well, on the left side, we have nitrogen attached to 2 hydrogens. We call this an amino group. And then if we look on the right side, we have what's called a carboxyl group. And so basically, every amino acid is going to have these 3 similar parts. And so the only thing that's going to be different is going to be what comes off the bottom, and we call that the R group. And so all amino acids are the same, except what comes off here, and that gives it different properties. And so let's kind of see how they're put together. And so when I build proteins inside my body, I'm doing that with amino acids, and we use that as something called a dehydration synthesis to do that. And so let me move this one over here. Basically what we do is we position one amino acid right next to an amino acid. You can see here that there is 2 hydrogens and an oxygen here. And you know that in chemistry, if we have 2 hydrogens, H2O, that's simply a water. And so what we can do is we can lose that water. Now it's not as simple as that. This whole thing sits inside a ribosome, so there's a giant enzyme around the outside of it. But let's attach another one. So now we bring another one right next to it. You can see that the hydroxyl group or the carboxyl group is attaching into the hydrogen. We're going to lose a water, and then we're going to form another covalent bond. And then we put another one next to it. We lose another water. We form a covalent bond. And we do that again. And now we have...
Those are called peptide bonds. Did I say that? The bond between a carbon and a nitrogen?
What's called a polypeptide. So each of these individually are called a peptide, but if we attach them all together, we have a polypeptide. And you can see that this strand across the top looks the same. It looks uniform, but the only thing that's going to be different in each of these is going to be the R group that trails off the bottom. So where does this occur? Well, basically, these are the amino acids. This would be the ribosome in a cell, and all life does this. Basically, you have these little tRNAs that will bring their amino acids in, and then we attach them together. And when you have a bunch of amino acids attached together, you have a polypeptide, and that polypeptide will eventually fold into a protein. And so here's our 5 amino acid sequence right here. These are each going to have different chemical properties. And so, for example, this one right here, threonine, is going to be polar. What that means is it's going to have a charge. If we look at alanine right down the way, this is going to be nonpolar. And so why is that important? Well, if you're polar, then you're hydrophilic. That means that water is going to be attracted to you. In other words, we're going to find threonine in the presence of water, but alanine, since it's got this methyl group right here, it's not going to be attracted to the water, and so it's going to hide from the water. Or if we look at the aspartic acid, it's going to have a negative charge, and if we look at the lysine over here, it's going to have a positive charge. And so these 2 things, or these 2 amino acids are found in the same polypeptide. They're going to try to get next to each other because the positive and the negative are going to attract. And so what you end up getting is a 3-dimensional protein. The middle part, so this brownish tan part in the middle, is going to be the backbone, and that's going to be, again, made up of